Say yes again.
Yes.
Cool, thanks guys.
So everybody prepared to do a whole bunch of writing?
Here's the example of the chapters 1 through 7.
I can't remember. What's in the...
Well, I only have it in chapters.
Where's mine? I have this class for after the class is finished, in case we run out of time. Okay? And I always say, you know, does anybody want to leave? No one ever does. So it goes through chapter... Okay, well, I'll try and get it done. You think I don't want to get it done? 1, 2, 3, 5, 6, 7, 9, and 10. Those are the chapters. No, the syllabus is on.
Okay, I see.
Yeah.
Okay.
1, 2, 3, 5, 6, 7, 9, and 10. Yeah.
1, 2, 3, 5, 6, 7, 8, 9.
Okay. Okay, I'll just make it one chapter. How's that? Now, I am not going to tell you what's going to be on the exam, okay? I'm not going to stand up here and tell you what the questions are on the exam. Much as you'd like me to do that.
Is it multiple choice? Like, what's the format of it?
Yeah, multiple choice and, oh yeah, that's the other thing. You need to go to the testing centre. Does everybody know where that is on Monday? It's above the lab. You know where the lab is? It's in the same building. Yeah. Yeah, the bowl. Yeah. Okay, and you know there's that circular staircase. You take that circular staircase, and at the top of that is the testing centre.
Second one.
Yeah.
Do you know the number of the...
There are 60. It's worth 150 points.
There's a big double door that says bowl.
It says testing center on it. Yeah. Multiple changes.
Do we need like a scan shot or anything?
No, because you're doing it on the computer. Yeah. Yeah, we're not coming here. Do not come here. Okay, go straight to the, at 2 o'clock on Monday, go straight to the testing centre.
How many questions are there?
60.
Are we going to go back, like after we finish it, can we run through, double check our answers, or is that not something?
Yeah, no, you can do that.
Is it through Canvas?
Yeah.
Oh.
Yeah.
Yeah, you can go back and you can
Do we still have labs?
You can scroll up and down. Yeah, you can scroll up and down. You don't just get one chance at the questions. You will know your results right away, but I can't let you see what you've done wrong because Canvas, it'll wake everybody up. Canvas makes it too difficult for that. But if you want to know, you can each come and see me, you know, in private, you know, during office hours, and I'll go through what you did wrong in your exam. You're allowed to look at it once, okay? You can't take notes though. Okay, so first of all, first exam review. Everybody got it so far? You need to know the difference between prokaryotes and eukaryotes. What's the main difference between them? Oh my God.
One has organelles and one doesn't.
Well, yeah, but what is the main difference? Chance just said it.
What is that? Nucleus.
Yeah, nucleus. Prokaryotes don't have a true nucleus, right? They don't have a nucleus. Okay, with the history of microbiology, you need to know the main players. You know, the ones I went through. But I asked you some of those questions in that first quiz, didn't I?
Mm-hmm. So can we use the ones mentioned in the notes as...
Good idea.
I just mean like there's...
I'm not going to ask you anything on the exam that is not in my PowerPoints. Got it? Okay. So,
If you look at the study guide, it's all on there.
Just,
yeah. I'm not going to ask you, you need to go through the PowerPoints. I'm not going to ask you anything. I've tried to make sure everything's on the study guide, but I'll go through it again just to be on the safe side. I just haven't had time, so far. I spent half the day yesterday working on the stupid exam because I hate Canvas. Let me tell you, I'll tell you again, I hate Canvas. And I can tell you again, I hate Canvas. I was just about in tears over stupid Canvas yesterday. And I went over to IT, I went over to Open Canvas. She said, you shouldn't take that attitude that you hate Canvas. That's the wrong attitude, because then it will be even worse for you. So, still hate it.
Thanks a lot.
Okay, so in class, remember, in the quiz, I think it was in the quiz, a lot of you got wrong that it was Van Leeuwenhoek who was the first person to see live cells, right? Robert Hope saw dead cells. It was Van Leeuwenhoek, the one that's hard to write. Yeah, the L word. The good thing about multiple choice is you just have to be able to recognise it, don't you? Okay, spontaneous generation. Some of you got that wrong too. Spontaneous generation, it's living organisms. They used to believe that living organisms came from non-living matter. Right? Is that true? No, they were idiots. So, who disproved that? Yes, Louis Pasteur. Everybody got that? Louis Pasteur. Do I have to write that on the board? Was that yes?
Yeah.
You want me to write Pasteur? Pasteur. Okay, Louis Pasteur. Who pioneered aseptic surgery?
That's the Nightingale, right?
No, that was Nightingale. Yeah, she wanted to keep everything clean, but it was a surgeon.
Joseph Lister.
Yes, Lister. Lister. Okay? And he used phenol. So that should, if you remember that he used phenol, you should know that phenol kills bugs, right? Kills microorganisms. It's got phenol in it? Oh, that's why it's called Listerine, because of Joseph Lister. That's exactly right. Okay, no Koch's postulates. Koch's postulates. It was a series of 4 steps, right? 4 steps. And the basic premise of it was that you could show that a specific organism caused a specific disease. They still use Koch's postulates today. So the first thing was you have to be able to isolate the microorganism from the animal that has the disease. You have to isolate it and grow it in pure culture. Then you have to be able to give the organism to a healthy animal and show that that animal gets the same disease. And then you have to isolate the organism from the second poor animal that was originally healthy and show that, and grow that in pure culture and show that that's the same microorganism as that you isolated from the first sick person, right? Yeah. So it's basically 4 steps. It's 4 steps, and you do the same thing twice, right? Everybody got it?
got it.
So, but you need to know the bottom line of it is to show that an organism causes a disease, that microorganisms cause disease, okay? The first vaccine. Who made the first vaccine? He did it on that poor little boy.
Jenner.
Yep, Edward Jenner. And it was the smallpox vaccine, right? Smallpox. Edward Jenner, and he did it on poor little Phipps. The little boy. Well, it saved him from getting smallpox, but it was a bit risky, wasn't it? These days you wouldn't be allowed to do it. The first antibiotic. What was the first antibiotic? Nope. Penicillin. The drug used for treating syphilis is actually not an antibiotic. It's a chemical. Yeah, penicillin is an antibiotic. And it was, who did that?
Fleming.
Yeah, Alexander Fleming. He called it an unfortunate, a fortunate accident because his cultures, his bacterial plates had got contaminated with a fungus, and he happened to notice that the bacteria, the colonies, were inhibited around the fungus, right? So he figured that the fungus had something in it that inhibited the growth of the bacteria, and he was right. That fungus makes penicillin. It's a penicillin mold, basically. Okay? So that was Alexander Fleming. Okay, macromolecules. Don't you just love chemistry? I'm going to make this as funny as possible because people say if I make it funny, well, I try to make it funny, they remember it better. So I guess the worse my jokes are, the better you remember it. So, the 3 types of bonds. What are the 3 types of bonds?
Covalent, non-covalent, hydrogen.
Covalent, ionic, hydrogen. Right?
Yeah.
Now, these are bonds between atoms within a molecule. Does everybody get that? Between the atoms within a molecule. Okay? Which is the strongest? I've written them in order of strength. This is the strongest, this is the second strongest, and this is the third strongest. Right? Covalent bonds. What type of bonds are they?
Polar, non-polar.
Yeah, you can have polar and non-polar, but you're making it way too complicated.
Electron
Covalent
sharing.
is sharing electrons.
Yeah, they're sharing electrons. That's why they're the strongest, because the electron shells actually overlap. Okay? Ionic bonds are between ions. What are ions?
Positively or negatively charged.
Yeah, they're charged atoms. And the reason that they've become, or charged molecules, the reason they've become charged is, I can put to the periodic table here. Where's my... Because remember, atoms are happiest when they've got full outer shells. They're not happy. They feel like shit. Oh, sorry. They feel bad when their outer shells, and she's recording it. Please don't repeat that. No. They feel bad when their outside shells are not full. They like to have their outside shells full. So if their outside shell only has a few electrons on it, they get rid of them. If it is only missing like 1 or 2, then they grab them from somebody else, right? So when they grab an electron, then they have more electrons in their outside. Remember, the electrons are on the outside and the protons, which are positively charged, are on the inside, right? Protons and neutrons. So if they grab an electron from somebody else, then they'll have an extra electron, so they'll have a net charge of -1. Because they normally have the same number of electrons as protons. So that way they're uncharged. So if they grab an electron from someone else, they're going to end up having a net negative charge. If they grab 2 electrons, they're going to have 2 minus charge. If they lose an electron, where is their eraser? Here.
There's one right here.
It's all right, got one. If they lose an electron, then they end up with a positive charge. So an ion is not the thing we're talking about. An ion is not the thing that you iron your clothes with. It's basically a charged molecule. And you know how like charges attract each other? Positive ions are attracted to negative ions. And that's the ionic bond. It's a bond between charged molecules of opposite charges, right? Hydrogen bonds. That's because in the water molecule, Okay, and the water molecule, remember oxygen is an electron hogger, right? It's greedy. And it's sharing, these are, now don't get confused about that. Within the water molecule, the bonds are covalent, right? Between the hydrogen and the oxygen, these are covalent bonds, right? Because oxygen is so greedy and keeps the electrons mainly to it, the oxygen part of the atom becomes negative, slightly negatively charged, and the hydrogens become slightly positively charged. So water molecules are attracted, the negative part of the water molecule is attracted to the hydrogens of another molecule, water molecule. So they're weak bonds between hydrogens and other molecules, okay? So the bonds between water molecules are hydrogen. The bonds within the water molecule are covalent. Yeah, don't get it mixed up. Everybody got it? Within, covalent. Between, hydrogen. Everybody got that in their brains? I'll try and zap it in there.
One more time. Okay.
Between, hydrogen.
Okay.
Within, covalent. We're only talking about water here. Okay? Got it?
Yes.
Okay. We've done a lot already. Okay. Composition of a buffer. I hate buffers. Buffer. How do you say buffer?
Buffer?
What's the difference?
Buffer.
I can't tell the difference. I think I sound like you guys. I've been here so long. I guess I don't. You know, when I hear myself recorded, I'm like, oh my God, what an awful New Zealand accent. Otherwise, how I hear myself? I hear myself as an American.
That's funny.
You don't hear me as an American, but I do. Okay. A buffer. A buffer consists of a weak acid and its conjugate base. I'll move away so you can see. Okay? What's that?
The other way around too.
Yeah. Can you see now? Weak acid and its conjugate base. It basically is something that can suck up hydrogen ions or suck up hydroxyl ions. So this part can bind to hydrogen ions and this part can bind to hydroxyl ions. So it keeps the pH stable. Because remember, pH, most organisms live at around neutral pH, well, a certain pH. And they can't live beyond that pH range. So they need buffers to keep the pH in their bodies or in their cells neutral or at a certain range. Okay? Okay. Aren't you just going to love this? You need to know how to do pH differences. For example, lemon juice. There's a pH of 2 and wine at pH of 4. Wine. Okay, which one is more acidic?
Lemon juice.
Lemon juice. Which one has more hydrogen ions? Lemon juice. Yeah. So the more acidic, the more hydrogen ions it has. Everybody got that? The more acidic, the more hydrogen ions. So lemon juice at a pH of 2 is 100 times more acidic, has 100 times more hydrogen ions than wine at a pH of 4.
Because each is with 10.
Right. Does everybody see that? Can everybody do that? If you can't do them, do the pH problems that I put online, with the answers. Go through them. Okay?
And we have to know the notation for it, which is .00 times What?
I'm just going to show you how to do a pH, actually work out the pH. So the pH equals minus log of the hydrogen ion concentration, right? Those square brackets just mean concentration, and P means log of, okay? So if the concentration is 10 to the minus 4, the pH would be, so the log of 10 to the minus 4, what's the log of 10 to the minus 4?
0.00.
It's minus 4. Yeah. So remember, this is why it's, so it is minus 4, so the pH is, yeah, pH is 4. Can everybody see how to do that?
So your log has a base of 10, which
Right.
cancels the 10 to the negative 4 out.
Yeah, so the log of 10 to the minus 4 is just minus 4. Yeah.
Log means your, what's it called, exponent.
Yeah,
right? It
it's
just
the
moves it down.
exponent. Yeah, the log is the exponent, exactly. And that's why this minus here is to make the pH a positive number. Because when you have a minus outside the brackets, you know, it turns that into a plus, right? Everybody know how to do that? And the hydrogen, if you want to do it the other way around, a pH equals 2, the hydrogen ion concentration would be 10 to the minus 2 molar. Well, it's actually, molar means moles per litre. Does everybody see that? Got to be able to do it both ways. Okay, functional groups. What everybody loves. Functional groups. I hope everybody looked at their quiz and they know what a phosphate functional group is. Can't remember what else goes over there, but it's just the one with the P and, yeah. Well, the R is the rest of the molecule. The functional group is this part. Okay? So what is this? What functional group is that?
Is that an amino?
Mm-hmm. It's an amino group. Yeah, if it has nitrogen in it, it's an amino group, right? NH2 is the amino group. This is the amino group part. Okay.
One's the carboxyl.
What's that?
Is the other one the carboxyl?
No, I'm going to show you. This is a carboxyl group.
I think you're thinking of the carbonyl.
So what is yours?
Carboxyl group is this.
The other one's the
Carbonyl. Yeah, carbonyl is, this is a ketone. Aldehyde is that.
Where do you find the list of the functional groups?
I put it in the lecture, didn't
It's in
I?
the slides.
Yeah, there's a PowerPoint of it.
Yeah, it's a whole table. It's exactly.
But
what
there's
it's
also
textbook.
there's also a page in your textbook, isn't there?
It's the same thing.
Yeah, it's just the same thing. Yeah. Okay. Now, what is this?
amino to the left, carbonyl to the right.
Bond.
It's an amino group there, and you've got a carboxyl group there.
Amino acid.
Yes! It's an amino acid. That is the structure of an amino acid. Each amino acid has a different group here. There's only 21 of them.
That's what I thought you were starting to draw over here. That's why I got
Yeah,
confused.
yeah. There's a carboxyl group and an amino group. Can everybody see that? What is the chemical reaction that joins smaller molecules to make bigger molecules? Yes, dehydration synthesis.
What was the question?
The chemical reaction that joins smaller molecules to make bigger molecules. You know, the monomers are joined together by dehydration synthesis, right? What's another name for dehydration synthesis?
Dehydration.
Condensation, because a water molecule is taken out, right? When it happens. An example of dehydration synthesis is this. Fructose plus glucose. If you join the two of them together, what do you get? Who's a good chemist in here? No.
It's corn syrup.
Sucrose, yes!
Isn't there only like 3 and then you combine them like in different ones and they make different sugars?
Well, there's all different sugars. This is a sugar molecule, this is a sugar molecule, and this is a sugar molecule that consists of a disaccharide. 2 sugars joined together. Each of these sugars, each of these here are called monomers. These are the monomers, they're monosaccharides. And this is a disaccharide. So what type of molecules are these? What's the name for them? We talked about 4 types of molecules.
Carbohydrates.
Carbohydrates! Yeah, they all have this formula, right? C2O, right? They have 2 hydrogens for every carbon and oxygen. Like glucose and fructose both have the formula C6H12O6. They're different because they're arranged differently, but they actually have the same formula, chemical formula. Okay? Remember, sugar molecules look like this. They're rings. Oops. Oh. Down. Up, down. It goes like that. They're in rings. The one in DNA is a 5 carbon ring. It has 5. It's a pentamer as opposed to a hexamer. These are hexamers. Okay. So the monomer of each, we did 4 molecules, right? 4 organic molecules. We did carbohydrates, and the monomer for a carbohydrate is?
Monosaccharide.
Yes, monosaccharide. That's the monomer. And examples of those monosaccharides are glucose, fructose. We did one in the lab, mannitol. You know, they're all monosaccharides. When they're joined together, big long strings, it's a polysaccharide, right? It's a polymer. It's a big long string. And they join together, the reaction that joins them together is dehydration synthesis.
Oh, yeah.
Yeah. Remember, when you're joining monomers together, the reaction is dehydration synthesis. The opposite, which occurs in glycolysis, is when you're pulling them apart, hydrolysis. Hydrolysis. Everybody got it? Dehydration synthesis, joining together, hydrolysis, pulling apart. When you join them together, you release water. When you're pulling them apart, it requires water. You have to put the water back in. That's what's called hydrolysis. Okay, lipids. What's the monomer of lipids? Fatty acid. Fatty acid. Yeah. The monomer of lipids is fatty acid. Remember that lipids are composed of a phosphate, the ones we talked about, the phospholipids. Lipids are often composed of fatty acids. Triglycerides which, have 3 fatty acids joined to glycerol. So you have glycerol here. And then you have your fatty acids hanging down here, right? Phospholipids, they have this fatty acid substituted with a phosphate group, right? You know, and remember you have the phosphate is the charged part, and then the 2 fatty acids, you know, they're like the hanging down things. They're the things that are hydrophobic because they hate water. Hydrophobic means hating water. Hydrophilic, liking water. So the phosphates, because they're charged, they're attracted to the water molecules because remember, each water molecule has a slight charge. You know, their water molecules are polar molecules because the oxygens are slightly negatively charged and the hydrogens are slightly positively charged. That's how water forms its solution type characteristic. You know how it's a liquid.
So the phosphate heads are hydrophilic?
Yes. And the fatty acids are hydrophobic. So they form away from water. Okay? Proteins. What's the monomer in a protein? I drew a picture of it. Amino acid. Yes. Amino acid. Remember I drew a picture of one. Okay? And how are the amino acids joined together? What's the reaction that joins them together?
Dehydration synthesis.
Yes! Dehydration synthesis! What's the reaction that pulls them apart? Hydrolysis. Yay! Now with proteins, remember there's 4 types of structure, right? There's the sequence of amino acids. What determines the sequence of amino acids? DNA. The genes. Yeah. The DNA determines the sequence of amino acids. Okay, so the primary structure of proteins is the sequence of amino acids. And they're in a string, right? The secondary structure is the alpha helices and beta pleated sheets. So it's this structure that's an alpha helices, and a beta pleated sheet is this type of thing. Okay? So that's the secondary structure. The tertiary structure is when the whole thing, you know, you have some alpha helices, some beta pleated sheets, and some globular stuff, and it's folded all, you know, into a glob, basically, with its active site, right? And the quaternary structure, so this is the tertiary structure, the quaternary structure, and not all proteins have quaternary structure. If they have quaternary structure, it's when it has more than one subunit. For example, hemoglobin has 4 subunits, you know, and it has a molecule of iron in the middle that binds to these, that's joined to these, holds them together. Okay, what determines the secondary and tertiary structures?
Disulfide bridges and hydrogen bonds.
Yes, but what determines where those are?
Primary structure.
Yes, the primary structure. Yeah, what holds, that's a good point, what holds the structure together?
The disulfide bridges.
The hydrogen bonds. Hydrogen bonds, but they also have some ionic bonds and have hydrophobic interactions. But what holds the glob together is the disulfide bridges. That's from the amino acids methionine and cysteine, because they have sulfur. They have sulfur atoms in their side chain. So the primary structure, the primary amino acid sequence, that was one of your quiz questions. No, that was one of your quiz questions, or one of your, I can't remember. I put it in something. Determines everything else, right? Yeah, it was a quiz. But I gave you the answers, right? So,
What determines everything else? The primary structure?
yeah. And what determines the primary structure? DNA. You guys are doing good. Okay, nucleic acids. What's the monomer of nucleic acids? Nucleotide, yay! And what are the two types of nucleic acids?
Purines and pyrimidines.
No, I'm talking about nucleic acids. Yeah.
Adenine, thymine.
No, those are the bases.
Okay.
DNA and RNA.
Oh.
Yeah, nucleic acids. I'm talking about the big things.
The big molecule.
Yeah, the big molecules, right? So the monomer is the nucleotide, and you need to know the structure of the nucleotide, the basic structure. So it consists of, the nucleotide consists of 3 parts. What are those three parts? You have the base. This is the base. It's actually a nitrogenous base.
Phosphate group.
Yeah. What's joined to the nitrogenous base?
Sugar.
Yep, the sugar molecule. And in DNA it is,
Deoxyribose.
yep, deoxyribose, right? So, deoxyribose. And in RNA it's ribose, right? And it's just different by this. In DNA there's just an H here, and in RNA there's an OH there. So this is the sugar molecule, which is deoxyribose or ribose, and then what's joined to that? The phosphate. Okay, so this is, which end of DNA is this?
5.
5' end. This is the 5' end. Where's the 3' end?
The circle on the
Yeah. The carbon on the deoxyribose or the ribose. This is the 3' end. Okay? So where the phosphate is the 5' end, and where the 3' end is where on the sugar molecule. Deoxyribose or the ribose. Okay? So you need to know what is joined to what. Okay? If I said to you, what's joined to the phosphate? You know that it's the deoxyribose. If I asked you, what is joined to the base? Yeah, or ribose. Depending, make sure you don't confuse the two. If it's DNA, it's deoxyribose. If it's RNA, it's ribose. Okay. Okay. Structure. The prokaryote, eukaryote flagella. There's fundamental differences in the flagella, right? The flagella of prokaryotes, how does it work?
Rotates.
Yeah, it rotates. Like a corkscrew. Yeah. And what drives it?
Sodium and potassium escaping the cell membrane.
It's actually hydrogens coming in. But what's it called? What's that called? It's the battery that drives it. We're talking about in prokaryotes and bacteria. The flagella is driven by the proton motive force. The proton motive force. What sets up the proton motive force? It's in the membrane. It's in the cell membrane. The electron transport chain. The electron transport chain makes the proton motive force. So there has to be an electron transport chain to make this, right? And you only get an electron transport chain in respiration, right?
Is the proton motive force the same thing as the gradient?
Yes, exactly. You know, because during the electron transport chain, the ETC, remember these things, cytochromes are set up here and electrons are passed down from one cytochrome to another, right? And as they do that, hydrogens are passed to the outside, right? So you end up getting a and water molecules are split, so hydrogen's past the outside and OHs are kept on the inside. So you end up getting a gradient of hydrogens on the outside and OHs on the inside. And the last electron acceptor, if it's aerobic respiration, is...
Oxygen.
Yes! Oxygen! Remember, because oxygen loves electrons. It's what it does in water molecule. It sucks up the electrons. It hogs them. So this is how the proton motive force is made. The battery is made. It's made during the electron transport chain, right? And that's what drives a bacterial or prokaryotic flagella. Okay, everybody knows what a prokaryote is, right? I'm just thinking, maybe I've jumped the gun a bit. Okay, so, but eukaryotic flagella are different. They don't rotate. They flick, right? They undulate. And they're not driven by the proton motive force. And don't get, bacteria have fimbriae, little hair-like projections. Eukaryotes have those hair-like projections, but they're called cilia. But in eukaryotes, they're involved in movement. In prokaryotes, they're not involved in movement. The only thing that's involved in movement is the flagella in prokaryotes. They're involved in sticking. The fimbriae, the little hair-like things on bacteria, they're involved in sticking to things. They're involved in adherence. They're involved in forming biofilms, because that's how bacteria stick together to form biofilms. What's the other structure that's involved in sticking?
Did you say fimbriae?
I've said fimbriae, yep. Glycocalyx.
I'm looking at it right
Yeah. Glycocalyx, right? And there's two types of glycocalyx. Yep. We stain the capsule in the lab. If it's real, and the other one is the slime layer. If it's really organized, and compact, it's called a capsule. And that's what we stained in the lab. Well, we tried to. I remember it was Janisha and somebody else who got the capsule. You two.
I got it too.
It was all right.
Ebony, right? Yeah, Janisha and Ebony.
Then I had to find it again.
But what does a capsule protect bacteria from? Phagocytosis. Yeah. Well, being lice, but being eaten up.
Well, I mean, like from...
After it's eaten up, it prevents it from, yeah. No, the thing that prevents the cell from lysis, from exploding, from osmotic lysis, is the cell wall. Yeah, don't get confused. Yes?
Will a biofilm compose the bacteria that have capsule glycocalyx? Will that look different from the biofilms that are just slime layers?
Because the bacteria are all mixed together, it's just one big globular mass. You know what they look like. You see them everywhere.
Okay.
Yeah, it's globular because it's a whole bunch of different types of bacteria. And the slime layer, what's the main thing that the slime layer protects bacteria from?
Water?
From becoming dehydrated. Yeah, you had the right idea. Yeah.
Is all that going to be on the exam?
Of course.
Okay.
Would I be talking about it if it wasn't?
We're going like in circles. Sorry.
Well, I'm coming out with extra things too, because when you take the exam, it's going to be like that. It's all going to be scattered around. Your final exam is going to be like that, so I want you guys to get used to having everything all over the place. I'm trying to go in order, but it brings up other things, and so I start talking about them. Yeah, we're talking about structure now, so that's why we're talking about glycocalyx. Okay. The plasma membrane, what is it composed of? That was one of the questions in the metabolism worksheet.
Protein.
Yeah. Some of you just said phospholipids. It's phospholipids and proteins. And it self-assembles, right? Self-assembles. If you put it in water, it will just form that shape. Is the plasma involved? Does it have anything to do with the shape of the bacteria, the plasma membrane? No. Because if you peel away the cell wall, they all become spherical. Yeah. Okay. The cell wall. You need to know the different structures. Gram negatives. I don't feel like drawing this. Gram negatives consist of, I have to take my sweater off now. The gram negatives consist of, you have the lipopolysaccharide on the outside, right, which is another membrane, right? Then you have a thin layer of peptidoglycan, and then you have the cell membrane, right? That's a gram negative. Why does that stain gram negative when you stain it?
Because it contains negatively charged something oic acid.
That's a good idea. But the reason is because when you add the crystal violet and the iodine, that keeps the purple in there. But when you add the decolorizer, what happens is the membrane is dissolved because it's alcohol. It contains alcohol. So it dissolves the outer membrane, and because there's only a thin layer of peptidoglycan, the stain can't be retained. Okay? So that's why it loses its stain. When you add the decolorizer, it turns clear. You need to know what colour they are at each stage. So a gram negative after the crystal violet is going to be purple. A gram negative after the iodine is going to be purple, right? When you add the decolorizer, what colour is the gram negative going to be?
Clear.
Clear, right, colourless. It's not going to turn red until you add the saffron in.
And what does it dissolve?
The outer membrane.
Oh, okay.
Yeah.
The LPS.
Yeah. Which the outer membrane is a bilayer, membrane bilayer, which contains LPS. And contained in that lipopolysaccharide, there's a very toxic molecule. What's that called? Write that just a bit. Yeah, endotoxin, which causes inflammation, septic shock. Endotoxin. It's the lipid A molecule.
Oh, yeah.
Yeah. The lipid A part of lipopolysaccharide is endotoxin, and it's released when the cells die. Okay? Endotoxin is everywhere. It's floating around everywhere. All drugs have to be tested for endotoxin before you can give them to people. Yes.
Sorry, I know someone just asked this.
That's okay, I don't mind.
It's destroying the LPS or just the
LPS, yeah.
Okay.
And then this, because it's not very thick, the stain comes out of the peptidoglycan.
Okay.
Okay? Does that make sense?
Mm-hmm.
So a gram positive would have a thicker layer?
Yeah, gram positives, this is the cell membrane, and then they have, remember, a thick layer of peptidoglycan. So you can see why they retain the purple colour. They retain the crystal violet because the peptidoglycan protects it from being dissolved. There's no lipid to dissolve, and so the peptidoglycan keeps the stain in there. But you can, if you over-decolorize it, you can still make the stain red if you leave your alcohol on too long. Okay? So know the differences between them. Know what colour each one is at each stage of the staining procedure.
Do they both have the space in between?
No. Only the gram negatives have this periplasmic space in between. Yeah, that's what it's called. In between the cell membrane and the peptidoglycan, and in between the peptidoglycan and the outer membrane. This is the outer membrane, right? The outer membrane is the outside membrane of gram-negatives. Okay, which microorganisms do not have a cell wall? There's one type of bacteria that do not have a cell wall. Yeah.
Microbacteria?
She's very close.
Mycoplasma.
Mycoplasma.
Oh,
Mycoplasma. Don't get it confused with mycobacteria. Mycobacteria are acid fast. They also have an unusual cell wall, mycobacteria. That's what causes tuberculosis. Mycoplasma, yeah.
Mycoplasma is like fungus, right?
No. No. Mycoplasma is a bacteria. Mycoplasma pneumoniae causes... walking pneumonias. What everybody gets, what's passed around campus all the time. You get a cough, a dry cough for 3 weeks. You're not sick enough to go to the doctor, but you feel crummy. That's why it's called walking pneumonia, and it's very infectious.
And it has no cell wall?
No. No cell wall. Right. Mycoplasma is the one that doesn't have a cell wall. Mycobacteria is the one that has an abnormal cell wall that you have to stain with an acid-fast stain, right? Acid-fast. Mycobacteria, acid-fast. Mycoplasma, no cell wall.
What does myco mean?
God knows. Maybe it's little.
Myco meant fungus. Like...
Mycology is fungus. Yeah, mycology is.
Yeah.
But these aren't... I think that they're called that because originally they looked a bit like fungus and they thought they were fungus. Yeah. But don't get confused. It's not fungus. Not fungus. It's a bacteria. Okay, they're very little bacteria. Very little. They form very tiny little colonies. Fried egg-like colonies. You don't have to know that till next part. Well, so you're going to need to know it soon. I don't want to overload your brains right now. Okay. What does penicillin do in the cell wall? Oh, yeah. What's peptidoglycan? Most people got those 3 questions right.
Does the penicillin break down the peptide?
Yeah. Penicillin. No, but first of all, what breaks apart the molecules in the peptidoglycan? Pretty much all of you got that right. Lysozyme. Yeah. Remember, it consists of N-acetylmuramic acid and N-acetylglucosamine. Now, you know, these are joined together one after another. That dimer, it's a polysaccharide. And lysozyme breaks that apart. And we have lysozyme in all our secretions.
And so it breaks down the cells?
It breaks apart the peptidoglycan. So it breaks apart the cell walls. Lysozyme is more effective on gram-positives than gram-negatives.
I thought that's what penicillin did.
Yeah, penicillin breaks apart the peptide bonds that join this together.
Which one keeps them from forming?
Penicillin.
So it doesn't...
It doesn't actually break the peptide bonds, it inhibits the formation of the peptide bonds. That's how it works. Just remember that it damages the peptide bonds, okay? And lysozyme breaks the strings. Is that an easy way to remember it? Lysozyme breaks the strings, penicillin breaks the peptide links between the strings. Everybody remember that?
No.
Penicillin actually prevents them from forming, but as long as you get the right idea...
Okay, if the peptidoglycan was just a series of strings, you pictured a series of strings that are linear like that.
Yeah, it wouldn't do anything. They've got to be joined together to form the structure.
There's bonds between those that are horizontal.
The strings, yeah.
And that's what the penicillin keeps from forming.
Vertical bonds.
Okay.
Yeah, or you could say horizontal if you're talking about them.
Depends how you're looking.
Yeah. Yeah. You got the... Does everybody understand that? Yeah. Yeah. That was a question in the metabolism. I did those. Okay. What are inclusion bodies? Inclusion bodies. That's how bacteria store things. They store them as inclusions. Examples of inclusion bodies are magnetosomes, which is how they store iron. Magnetosomes. Lipids, they're just called lipid inclusions. Carbohydrate storage granules. Are ribosomes inclusion granules? No. Ribosomes are where proteins are made.
I have a weird question about an inclusion in anatomy. They taught me that the tattooing sits on the top of our cells as an inclusion. Is that correct?
I don't know. Probably. Sounds reasonable to me. Sounds good. But we're talking about inclusions inside bacteria. Okay. The different shapes of bacteria. 3 main shapes, right? Cocci, bacilli, spiral. Right? Cocci, bacilli, spiral. And there are 3 types of spirals. There's the comma, vibrio. There's the spirochete, which is flexible. Treponema pallidum and Borrelia burgdorferi are spirochetes. Right? And they have that special type of flagella, spirochetes. Axial filament, right? Everybody got that?
Do spiral ones have axial filaments?
Yeah, spirochetes, not spirillum.
The 3 were comma, spirochete, and what?
Spirillum, where it's a rigid spiral. Helicobacter pylori, which causes stomach ulcers, is a spirillum. Okay. Now, don't get the arrangements and the shapes of bacteria mixed up, okay? Remember, the shapes is the shape of the cell. The arrangement is how they're joined up to each other. So, yeah, strepto is a string, beads in a string. Staph is bunches, grape-like clusters. You can only get staph in cocci. And you can get strep in bacilli and cocci. You can't get spirals. Don't have any of those. For those who have to go, I'm just going to say, I'll tell you. Oh my God, we've got a bunch of here.
You got a lot more.
Quite a bit, yeah. Do you have to go? Oh.
Are you taking back the worksheet you said?
Oh yeah, let me give those back. Does anybody want to stay and keep working?
I'd love to, but I gotta go get my kids.
Here's the, I'll stay for as many people who want it. So here are all the
Yeah, I
worksheets.
have to be at work at 5, so I'm not
So you write my email down?
Yeah.
Or I'll write it down.
That one. I've corrected everything. Do you want yours, Hayley? Do you want yours? This is, oh, Edie. Beautiful. I'll come to yours. Can you see yours, Hayley? Tell me I didn't bring it.
See mine right now.
There.
Thank you.
Make sure you read all your questions properly for the exam, okay? Because you're getting everything right, you're just missing it. Okay.
What, like maybe another half hour?
What's that?
Maybe like another half hour?
I hope so, yeah. Yeah.
I'm gonna sit in the back because I like have to.
Yeah,
Now I go ahead and get mine too, because I might have to dart out also.
right.
Then we don't have to disturb you again for that. You want this?
Yeah.
Can you give me Chris's also?
You.
Mine's here.
Oh, ma'am, this one.
Tanisha left. Okay, all the rest are here.
You don't get perfect scores, do you?
What's that?
You don't get perfect scores, do you?
Yeah, I do. I've given a whole bunch of perfect scores for the last lab reports.
I got so perfect scores.
There, he got a 5 out of 5. Sorry, I'm not supposed to say what other people got.
Can we have ours too? Do you want me to get them or what do you need?
Stephanie. Not sure. Yeah, she got a little bit wrong, but I rounded everything up.
9.9, okay.
Yeah, I rounded everything up, so you guys were lucky. Katie, right?
Thank you.
And you have sent yours in, right?
Was each question was worth 1 point?
No, it was worth, because there were parts, and it was out of 5, so, you know, it was out of 10, and then I put it out of 5.
So what did I get?
You got a slight bit
Oh,
here.
okay.
You said what it was, so I gave it to you. But you
Oh,
didn't
okay.
actually put allosteric, I don't think. Okay, shall I keep going? Yep.
That's what allosteric means, though, right? It's just a place other than the competitive site.
But it's actually called the allosteric
Okay.
site. Okay? Yeah, make sure you know the stuff that's on the quiz. Okay? Important. Okay. Know the different arrangements of flagella. A lot of you in the worksheet for I'm reminded of this, but you know when I asked for different oxygen requirements for bacteria and where you would find them in the tube, a lot of you only answered half of that. You would say either where they were found in the tube or what the oxygen requirements were. You didn't say both. So be careful that you read the whole questions. So when you're doing your exams, don't jump to the first conclusion, because there may be some that are like all of the above or something like that. Okay, so be careful. Yeah, know the different arrangements of flagella, but we've already talked about that. That was in the metabolism quiz.
It's in the worksheet too.
Yeah, in the worksheet. Yeah, that's why I put a lot of those things in there. Endospores. Know what endospores look like. Okay, in a cell. You have a cell like this.
How they look like when stained or just in general?
Yeah, in general. Not necessarily when stained. You'll see that inside a cell.
There's a really good picture on the
In other words,
the lab sheet or whatever.
Right, yeah.
With the pink.
Yeah. In other words, if you're confronted with something that's not stained, remember I've pointed them out to you guys because you've had cells with endospores in them. I've pointed them out to you. You can even see them when they're not stained. There'll be clear areas, circular areas within the cells. Okay? Does everybody, if you don't know, just look online. You'll see it. I'm sure there's pictures in the textbook too. Can a cell produce more than one endospore?
No.
No. Each cell only produces one endospore. And then, don't get confused with endospores and with fungus spores. Fungus spores are involved in reproduction, right? Endospores and bacteria are not involved in reproduction. They're involved in survival. The cell forms them when they run out of nutrients. There's only two types of cells that form them, and they're Clostridium and Bacillus species. I already said no, the gram stain. What is chemotaxis?
Movement or
Chemotaxis?
Yeah, chemotaxis. Taxis is movement towards and away from something. Yeah.
Towards a chemical?
Yeah, it's movement toward or away from a chemical. Yeah, very good. So know the different, competitive inhibition, non-competitive inhibition, right? Competitive inhibition, the competitor binds to the active site, right? That was in the worksheet. Non-competitive inhibition, it binds to the
Allosteric.
yeah, Mitchell's got it there. Allosteric site.
Sorry, that's me.
Enzyme, so know the enzyme structure. An apoenzyme is the non-active part. Remember some enzymes need cofactors or coenzymes to become active. When it's the complete enzyme, it's called a haloenzyme. Right? The apoenzyme is the non-active part of the enzyme. But not all enzymes need coenzymes. Apoenzyme, yeah. The haloenzyme is the complete apoenzyme. The apoenzyme plus the cofactor or the coenzyme makes the haloenzyme. Okay? Okay, glycolysis. What's the end product of glycolysis?
Acetyl-CoA.
No.
Pyruvic acid?
Yes! Hooray! Third time. Pyruvic acid. Pyruvic acid. The end product of glycolysis is pyruvic acid. How many ATP molecules are made in glycolysis? Two. Right. There's actually 4 that are made, but 2 are needed to get the reaction going.
So 2 are needed or 2 are made?
2 are used.
Used.
Okay.
There's actually 4 made altogether.
So how many does it yield?
2. Yeah.
It needs 2 and it yields 2?
It uses 2 and yields. Yeah, the net gain is 2. It makes 4 altogether, but 2 are used in the process, so the net gain is 2. Yeah.
Do you know the full Krebs cycle?
No. You need to know the bottom line, and that's what I'm telling you right now. Yeah. It helps if you know what's going on, because that helps you to understand it. So with glycolysis, you start out with glucose, and you're going to lyse that, right, because it's glycolysis, lysis. What happens, there's 6 carbons in glucose. What happens to all those carbons? That doesn't occur in glycolysis. Sorry, I'm jumping the gun. Okay. So we know what the basic, the end product of glycolysis is pyruvic acid. We know, what is the main purpose of ATP?
Energy, basically that.
To carry energy. I asked that question, I said, what is the main energy carrier within the cell in the worksheet, right? So many people didn't put ATP. It's ATP. ATP is the energy currency. Are you taking a picture of me?
No, it's recording.
Oh, good. I don't want to be photographed. I did have that happen to me other times. Not real big on being photographed. Okay, so Now, if the cell doesn't have any oxygen available, you know, the process is, yeah, fermentation, right?
So how would we word that? Absence of oxygen would be fermentation.
Well, you're not going to have to word it because these are multiple choice questions, right? So the products of fermentation are organic molecules such as lactic acid, ethanol. Can't remember the other ones. So if you have yeast, you know, yeast can undergo fermentation and respiration. If you have, we should go through the other parts first.
You said lactic acid.
Yeah, that's it.
Isn't that produced in our muscles?
Yes.
So how does
When
that-
they're fermenting. When you're exercising too much, when you when there's not, you're not getting enough oxygen. That's why they start stinging.
All right.
Is it true that grass can release that?
Lactic acid?
Mm-hmm. Like I've heard that, like
If you
truck
eat
runners,
grass?
like if you walk on the grass, like afterwards barefoot, that it helps release the lactic acid.
I don't know.
Sore.
Never heard of it. Sounds an interesting idea. Yeah. It's the lactic acid, you get the buildup of lactic acid, and that's what stings. Because, you know, you're puffing like crazy to get enough oxygen, but you can't get enough oxygen to your muscles. And they start fermenting and making lactic acid. Okay, respiration. Respiration involves the Krebs cycle and an electron transport chain. Right? In order for it to be called respiration, it has to include Krebs cycle and electron transport chain. Anaerobic respiration, oxygen is not the final electron acceptor. It's only called aerobic respiration if oxygen is the final electron acceptor in the electron transport chain. So, you have the Krebs cycle, right? Cycle, God. And then you have the ETC, right?
Is glycolysis considered a part of the Krebs cycle?
No.
It just comes before.
There's 3 parts of respiration, right? But this by itself is not respiration. In order for it to be respiration, you have to have all 3 parts, right? If oxygen is the final electron acceptor, then it's called aerobic respiration. If oxygen is not the final electron acceptor, then it's anaerobic respiration. So don't think just because it's called respiration, it always involves oxygen. It doesn't. Okay? So, what's the main function of the Krebs cycle? That was one of the questions on the metabolism worksheet.
To produce DH and
DH2.
FADH2 to carry the electrons.
Yeah, to feed into the electron transport chain. Because this is driven, the electron transport chain is driven by NADH and FADH2, right? In the Krebs cycle, what happens to all the carbon atoms from the glucose molecule? They're released as CO2. Right? We breathe those out. You get 6 molecules of CO2 produced. What happens in aerobic respiration if there's no oxygen?
Shut down.
You shut down all of this.
None of that can happen.
Yeah, none of it can happen. And for glycolysis to occur, you have to regenerate the NADH, the NAD. Because remember, there's one step in glycolysis that makes NADH. Okay? So this has to be turned back to NAD in order for glycolysis to keep occurring. So that's why you make lactic acid or ethanol. Those are made to regenerate this, regenerate the NAD. Okay, but no energy comes out of it. Now, during the electron transport chain, you get 3, if oxygen is the final electron acceptor, you get 3 ATPs for every NADH that goes in, and you get 2 ATPs for every FADH2. And when ATPs are made by the transfer of electrons, you know, what's that called? The making of ATP by the transfer of electrons. Oxidative phosphorylation. Does everybody remember that when I say it?
Yeah.
What's that? Oxidative phosphorylation is when ATP is made from the transfer of electrons. You know, from NADH and FADH2, right?
And it's called phosphorylation because of the 3-phosphate.
Well, because ATP is made. That's what's called phosphorylation. ADP is converted to ATP, right? Okay. ATP is made in glycolysis by substrate level phosphorylation because the phosphates are transferred from a substrate. What is the overall purpose of cellular respiration? That's probably way too complicated for you guys. Yes. It's to capture the energy in the bonds of glucose and convert that energy into ATP. Because it starts out with glucose, so you're capturing the energy that's in those covalent bonds, you're breaking them apart, and you're putting it into, you're transferring that energy into ATP. That's exactly what that process does. Which produces the most energy? Aerobic respiration, anaerobic respiration, or fermentation? Aerobic. Which is the next most? Anaerobic? Yep. Which is the least? Fermentation. Fermentation only makes 2 ATPs.
So anaerobic respiration is just when the glycolysis process continues?
Yeah, you still have the Krebs cycle and the electron transport chain, but the electron transport chain isn't as effective because oxygen is the best electron acceptor.
Right.
So, and sometimes you don't have the whole Krebs cycle. They use another molecule. They use another inorganic molecule to accept the electrons.
Oh, okay, so they just have different electrons.
Exactly.
And then the difference between that and fermentation is when it all gets shut down and they have to produce another chemical to
remove the hydrogens from NADH. Okay? Got it? Generation time. What's the generation time? We're nearly done. The time it takes for a cell to divide into 2. So time it takes for a population of cells to double.
2 by 2.
Yeah, but except it's mitosis because it's not mitosis because
Generation time.
generation time. It's also called the doubling time. It's not the time it takes. This is prokaryotes as opposed to eukaryotes because it's not mitosis. It's not the time it takes for the nucleus to divide. It's not the time it takes for the DNA to be replicated. It's the time it takes for one cell to turn into 2 cells.
Are those going to be the other options on the...
I'm not going to tell you that. So you need to know after 4 or 5 generations, if you start out with 1 cell, how many cells it would be. Something like that.
So we're going to be drawing some pictures.
Yeah, you can have note paper. So after 6 generations, if you start out with 1 cell, how many cells would there be?
Well, 6 squared, 36.
So the first generation is 1 to 2. The second generation is 2 to 4. So that's number 2. The third generation is 4 to 8. That would be number 3. The fourth generation is 8 to 16. The fifth generation is 16 to 32. So the sixth generation is 32 to 64. So with 1 cell, after 6 generations, you're going to have 64.
Oh, so 6 generations after the first cell, so you're not counting the first cell?
The first cell is generation 0.
Okay. Because see, I would have thought 32 would have been...
Because it's to the power of. So it's 2 to the power of 0 is 1. Okay? So it's to the power of, because it's, it's logarithmic, but to the power of 2, not to the power of 10. Do you see what I mean? I was trying to make it easy, though, so you don't have to do it by the math way. If you think about it like that, you can see what's going on, can't you? Okay. Know the growth curve. Okay, so this is the lag phase, this is the log phase or exponential phase, this is the stationary, and this is the death phase. Which phase is our logarithmic?
The log phase.
The exponential increase phase.
Yeah, the log phase and the death phase. They're both logarithmic. So cells die at a logarithmic rate, and this is when they, everything's great. They're basically at a smorgasbord, they're at a table and all food is available, and they're eating their hearts out and dividing like crazy. This is when they run out of nutrients. So the stationary phase is where the number of dividing cells equals the number of dying cells. So it's static.
So it's like your childhood, then puberty, then stationary is like your midlife crisis, then you die.
There you go. And this is when you're getting, this is before, this is childhood. They're getting used to the nutrients. It's before they know any better.
Do we need to be able to identify
Yes.
the chart?
Yes.
Okay.
Yes.
So there'll be pictures on this place?
I'm not going to, there may be. I'm not going to tell you that.
Not that picture, but pictures.
There may be lots of pictures.
Okay, that's good. That's
May not be. Okay, what achieves sterilization? How do you
For
sterilize?
this class, it'd be the thing.
Prions.
Autoclaving. Autoclaving achieves sterilization, but it doesn't get rid of prions, right?
Or through moist heat.
Yeah, it does get rid of endospores. Does. By definition, sterilization gets rid of everything, but autoclaving does get rid of everything, including spores, but it doesn't get rid of prions. Prions, you've got to use sodium hydroxide, right? Disinfection is the destruction of all the vegetative forms. Disinfection is destruction of vegetative forms. A vegetative cell is a cell that's not a spore, right? So the process of forming a spore is called sporulation. The process of a spore going back to a vegetative cell is called germination, right? Antisepsis is cleaning off. So disinfection is destructive of vegetative forms on an inanimate surface, like this table, right? Disinfection. Antisepsis is removal of all the vegetative forms on a living tissue.
Just vegetative?
Yeah.
What was it?
Antisepsis. Sanitization is reduction of microorganisms to a safe level. So it's not getting rid of them all. That's what the definition is.
What is it?
Sanitization.
What was the definition?
Reduction to a safe level. Okay, static. If something is static, like bacteria static or fungus static, you're inhibiting the growth. If it's cidal, you're killing it. It's history if it's cidal. So if it's bactericidal, kill them. Most antibiotics are bacteriostatic. They inhibit the growth so that your body's own defences can take control and get rid of it. What's phosphorus required for when we're talking about nutrients? What do you need phosphorus for? Membranes, yep. DNA membranes, right? Phospholipids. What is nitrogen required for?
Proteins.
Yes, proteins, DNA. I'll say it slower.
Okay, yeah, sorry, I'm just still trying to write what you were just saying, so I'm getting behind. Okay, so phosphorus, what was the next one?
Phosphorus, phospholipids, and DNA, right? ATP. Nitrogen, proteins, and DNA. And included in the RNA too, you know, all of the nucleic acids. Included in those are ATP, right? You can't just not include ATP. What's the limitation of the autoclave? Yeah, that's one. You can't use it for things that are heat labile. In other words, you can't use it for anything that's got proteins in it. You can't use it for plastics. What happens to plastics? Yeah, I remember, you know, when I was a graduate student, we had idiot graduate students, you know, who took so much pleasure in autoclaving all sorts of things. Like, you know, those big styrofoam boxes, they would autoclave them and they'd come out as little tiny styrofoam boxes. Too much time on their hands. Okay. Differences, need to know the differences between physical and chemical control. Make sure you know the differences. You know, chemicals involves chemicals. Physical is like heat, physically removing something, like washing your hands is physical, unless you've got chemicals in there.
Would water be considered a chemical, or?
I'm not going to ask you. You wouldn't use water to get rid of things, would you?
I mean, if you're just washing your hands with water.
Yeah, the physical action of it, but I'm not going to ask you that, so don't worry about it.
You said it's the difference between chemical and physical.
Ways of getting rid of bacteria. You know, microbial control. You know, there's a list. Look at the PowerPoints. Make sure you know the differences between them. Okay, what's a surgical scrub that's used? There's two types of surgical scrubs. Yeah, it contains iodine. What's it? Betadine. Betadine is just a long-acting iodine. And the other one is chlorhexidine.
Or hexidine.
Betadine. Surgical scrub. Betadine. It's in your PowerPoint. Chlorhexidine.
Aren't some people allergic to betadine?
Probably. But they are.
My mom's allergic to iodine.
Hmm?
I think my mom's allergic to iodine.
Yeah, and it contains iodine.
So does that mean she can't have salt that has iodine in it?
Iodized salt. You need iodine because otherwise you get a goiter, don't you? Okay, DNA synthesis. This might take me a little while, so I'll just try and say it as quick as I can. DNA synthesis involves the replication of DNA, okay? And remember, this is vertical transmission. This is copying your DNA in order to pass it to your daughter cells. You have to copy your DNA so that your genes are passed to your children, right? So the first thing before cells divide, the first thing they do is they copy their DNA, right? And the process of copying DNA is called... No, copying DNA into DNA. Replication. DNA replication. If you're having trouble with this stuff, look at the animations, okay? Look at the animations. And what's the enzyme that does this? The enzyme that copies DNA into DNA. Yeah, it's a polymerase, but which polymerase? DNA polymerase. DNA polymerase. Don't get mixed up with TAC and everything. I haven't told you about TAC yet. Okay, you're making it confusing. DNA polymerase is TAC polymerase is just a type of DNA polymerase.
That's the one that we isolated from bacteria.
That grow in high temperature.
Yeah,
okay? Don't make it confusing. DNA polymerase copies DNA into DNA. Now remember that the DNA replication in bacteria goes in both directions. It's bidirectional, so you have a replication fork going in both directions, right? At the beginning of the fork is the unwinding enzyme. It has to unwind the DNA. That's called the helicase.